Hello, my name is Izzy. So, as you can see here, the yellow sticky notes are the steps in order 1 through 7. They're going like a circular motion that you can see. And the blue sticky notes is the steps more in depth on cystic fibrosis, which Nada will be going over shortly. Step 1. Cells are removed from the patient. Step 2. In-lab virus altered so can't be reproduced. Step 3. Correct gene is packed in a vector which is inserted into the virus. And step 4. The altered virus is mixed with patient cells. And Destiny will pick up where I left off.
My name is Destiny, and I will be continuing the steps of gene therapy. In the 5th step, the cells start to become genetically altered. Next, the altered cells are injected back into the patient. In the last step, the altered cells will allow for the desired proteins to be produced. Next, Nada will explain the steps of gene therapy with treatment of cystic fibrosis.
I'm Nada Mustafa, and I'll be talking about how gene therapy helps with cystic fibrosis. So firstly, in cystic fibrosis, the CFTR gene is mutated, and that doesn't allow for the CFTR protein to open, which stops chloride ions from moving across the cell membrane. So chloride ions are what help get rid of and loosen the mucus. So without them, this leads to a buildup of thick mucus. With gene therapy, the correct CFTR gene is introduced to the cell, and the normal copy of the CFTR gene will also be present with the mutated gene. The normal CFTR gene will make a functioning CFTR protein, and that will allow for the chloride ions to move across the cell membrane. Since the mutated gene and the normal gene will be present at the same time, only some ions will be able to move across the cell membrane. This will help loosen up the mucus, and it will help with the cystic fibrosis symptoms, but it will not cure it, and there will be much less buildup in the end.